Thermodynamics tells us whether a reaction will occur, but knowing a reaction is spontaneous doesn't tell us anything about how fast the reaction occurs. Kinetics is the study of reaction rates. Reaction rates tell us how fast the reaction occurs. A reaction rate is the change in concentration of a reactant or product with time. Typical concentration units are molar, and typical time units are seconds. We can determine a reaction speed by measuring the rate at which it occurs. When A is converted to B, the concentration of A decreases, and the concentration of B increases at the same rate. Thermodynamics tells us that sugars, such as sucrose and glucose, decompose spontaneously. We know from personal experience that we get energy from eating sugar. When we eat sugar, it decomposes into water and carbon dioxide. How long will it take sugar to decompose into carbon dioxide and water? It depends on the conditions. If we heat the sugar, its decomposition will take just a few seconds. Sugar also won't take very long to decompose if we eat it. But if we leave it alone, sugar will take years to decompose. Why are the reaction rates so different? Let's use an energy diagram to answer this question. An energy diagram illustrates the energy pathway of a reaction. The products are at a lower energy than the reactants. This tells us that the change in free energy for the forward reaction is negative and the reaction is spontaneous. Notice the hill on the energy diagram. The hill represents an energy barrier, something the reacting molecules must cross in order to become products. The energy barrier is called the activation energy. The activation energy is the minimum amount of energy needed to initiate a reaction. At room temperature, very few of the glucose molecules have enough energy to cross this barrier. When heated, the kinetic energy of the glucose molecules increases and they react. What speeds up the reaction when we eat sugar? The reactants follow a pathway with a lower energy barrier. Catalysts in our bodies guide the reactants through this new course. A catalyst is a substance that increases the reaction rate without being consumed. The catalysts used by living organisms are enzymes. Notice that while the hill is lowered, enzymes do not affect the free energy values of either the reactants or the products. These values are related to how far the reaction proceeds before equilibrium is reached. Enzymes have no effect on the reaction equilibrium. When we need quick energy, we eat a candy bar. It's a good thing we don't have to wait years for the metabolism of sugar to occur. In this activity, we'll learn how enzymes work as catalysts, how they catalyze specific reactions, and how reaction conditions affect reaction rates. Are enzymes. Most enzymes are proteins. Proteins are polypeptides made up of amino acids. The polypeptides fold into secondary structures such as alpha helices and beta sheets. These secondary structures then fold into the protein's tertiary structure. The tertiary structure of a protein is important for its function as an enzyme. When a protein unfolds, this structure is lost, and the protein is no longer active. Specific regions of the enzyme molecule bind other molecules called substrates. Substrates are the molecules that react. In this example, glucose and ATP are the enzyme's substrates, and the enzyme is hexokinase. How do enzymes work? Enzymes catalyze reactions by lowering the activation energy. How do they lower the activation energy? Enzymes are relatively large molecules. They form many contact points with their substrates. The formation of the enzyme-substrate complex is the first step in an enzyme-catalyzed reaction. The enzyme holds the substrate in the proper orientation for a reaction to occur. It places the substrate near specific amino acids and cofactors that help it react. The contact points or binding interactions between the enzyme and substrate result from hydrogen bonds, van der Waals interactions, and ionic interactions. More enzyme-substrate binding interactions form once the substrate has started to react. This occurs at the top of the hill and is called an activated complex, or transition state. When an enzyme binds a substrate, energy is released. The binding energy lowers the activation energy. Because the hill is lower, the reaction occurs more quickly. Enzymes bind substrates and convert them to products. During the reaction, the enzymes are not changed. One enzyme can convert many substrate molecules into products. Now that we know what enzymes are and what they do, let's learn more about how they do it. Enzymes are very selective in the types of reactions they catalyze and the types of substrates they act on. Let's explore this concept of specificity by examining a biological process in which enzymes play a critical role, the pathway called glycolysis. Glycolysis is a series of 10 reactions that convert glucose to pyruvate. Pyruvate is an intermediate formed when glucose is metabolized to carbon dioxide and water. How do enzymes in this metabolic pathway know which reactions to catalyze? Let's focus on the first reaction in the glycolysis pathway. The enzyme hexokinase adds a phosphate to glucose. It doesn't add the phosphate just anywhere. It specifically adds it to the C6-hydroxyl. Other enzymes exist that phosphorylate the other hydroxyl groups. Hexokinase adds the phosphate to the C6-hydroxyl because of the way the enzyme specifically binds the glucose molecule. Hexokinase can't phosphorylate glucose directly because the free energy change is positive, so it couples glucose phosphorylation to ATP hydrolysis. To couple these reactions, hexokinase has a binding site for ATP and another for glucose. This is the enzyme's active site, where catalysis occurs. Catalysis is the change in the reaction rate by a catalyst. Because ATP hydrolysis is spontaneous, it doesn't need the glucose in order to react. It can react with water to form ADP. Water is smaller than glucose and easily enters the active site. Reacting ATP with water is like setting dollar bills on fire. You get a little bit of heat, but nothing else. Hexokinase must specifically catalyze the reaction of ATP with glucose and no other molecule. How does this occur? Hexokinase binds ATP only after glucose binds. This prevents the enzyme from catalyzing the reaction of ATP with water. The enzyme changes its shape after glucose binds. This is called induced fit. The shape change promotes ATP binding, thereby improving the enzymatic activity. The many points of contact between the enzyme and its substrate also ensure specificity. Molecules with slightly different shapes won't fit into the active site as well as the substrate. This reduces the ability of the substrate to interact with the enzyme, and the activation energy remains high. Often an enzyme can't do its job alone. Molecules called cofactors are needed. A cofactor is a molecule required for the action of certain enzymes. Cofactors may be inorganic ions such as iron or magnesium. The enzyme hexokinase requires a magnesium ion as a cofactor. Magnesium ions function to shield the negatively charged groups of ATP and facilitate the reaction with glucose. Coenzymes are also cofactors. A coenzyme is an organic molecule often derived from vitamins. Unlike an enzyme, a coenzyme may be modified in a reaction. The coenzymes NAD and NADP carry hydride ions in oxidation-reduction reactions. A hydride ion is equivalent to a proton and 2 electrons. Enzymes are the world's best catalysts. Industrial catalysts speed up reactions by 3 to 5 orders of magnitude. They often require high temperatures and they aren't very specific. Biological catalysts increase reaction rates by 9 to 15 orders of magnitude. They work at room temperature and are extremely specific. In the next section, we'll learn more about reaction rates when we discuss enzyme kinetics. Enzyme kinetics describe how reaction rates are measured. We can use enzyme kinetics to determine how long it takes an enzyme to convert a substrate into a product. Let's look at the conversion of hydrogen peroxide to water and molecular oxygen. Hydrogen peroxide is a strong oxidizer. It's used to clean scrapes and cuts. A bottle stored in a medicine cabinet lasts for a year or longer. Inorganic catalysts, such as iodide ions, speed up the reaction rate by about 5 orders of magnitude. Ever notice how hydrogen peroxide also bubbles when you pour it on a cut? A catalyst must be present. The enzyme catalase is released when cells are damaged. Catalase catalyzes the decomposition of hydrogen peroxide to water and oxygen. Catalase has 4 subunits, just like hemoglobin, and an iron-containing heme group. It also requires the coenzyme NADP. Catalase speeds up the decomposition of hydrogen peroxide by 15 orders of magnitude. Catalase can degrade 10 million molecules of hydrogen peroxide in 1 second. The reaction rate depends on how often an enzyme and substrate collide. The catalase reaction is limited by how fast the reacting molecules can diffuse and collide with the enzyme. Reaction rates depend on the reaction conditions. Since a reaction can't occur any faster than the time it takes for 2 molecules to collide, reaction rates are sensitive to concentration. The rate of an enzyme-catalyzed reaction will increase with concentration until the enzyme is saturated with substrate. When the enzyme is saturated, all active sites are filled with substrate. In general, only initial rates are used in kinetic calculations. The initial rate can be measured in this region of the left graph, which shows change in substrate concentration as a function of time. This is important because the substrate concentration decreases as the reaction proceeds, thus affecting the reaction rates whenever the enzyme is not saturated. Temperature also affects reaction rates. In general, the rate of a reaction increases as temperature increases. Recall the combustion of sugar. However, the reaction rates of enzyme-catalyzed reactions won't keep increasing forever. At high temperatures, proteins unfold and denature. Because an enzyme's catalytic ability depends on its structure and being able to form lots of substrate-binding interactions, the unfolded enzyme doesn't work very well. Many of the reactions that enzymes catalyze involve the transfer of protons. Proton transfer reactions are very sensitive to pH. Recall that pH is a function of proton concentration in the solution. A protein's structure is sensitive to pH. Extremes in pH cause proteins to unfold. Each enzyme has its own unique temperature and pH profile. Because of the charges on substrates and enzymes, ion concentrations influence reaction rates. The ions shield the charges to prevent proteins from aggregating and precipitating out of solution. In addition to environmental conditions, molecules called activators and inhibitors affect enzyme activity. Activators and inhibitors are molecules that bind to enzymes. Activators and inhibitors are like the volume controls on a stereo. Activators turn up the volume and improve the catalytic ability of enzymes. Inhibitors turn the volume and the enzyme's activity down or off. Some activators and inhibitors change the enzyme's shape. An enzyme can have conformations with different activities. Recall how hexokinase's shape changed upon glucose binding. Let's look at a dimeric enzyme that has 2 forms, an active form and an inactive form. When an inhibitor binds, the inactive form is favored. When an activator binds, the active form is more likely. Many metabolic pathways are regulated in this way. This is called allosteric regulation. These molecules, called effectors, bind at a site other than the active site. Another type of an inhibitor is a competitive inhibitor. Competitive inhibitors compete with the enzyme's substrate for the active site. An enzyme's activity depends on its structure. Its structure depends on temperature, pH, ion concentration, and effector concentration. Enzyme activity also depends on substrate concentration and the concentration of competitive inhibitors. Many drugs exert their therapeutic effects by acting as competitive inhibitors of target enzymes. Enzymes are the world's best catalysts. They work in water at relatively low temperatures and with extreme specificity. They cannot make an unfavorable reaction spontaneous, nor can they change how far a reaction proceeds. They can lower the energy barrier, which speeds up the reaction rate. Enzyme kinetics describe enzyme-catalyzed reaction rates. Energy is released when an enzyme binds its substrate. This energy is used to lower the activation energy. The lowering of the activation energy enables the reaction to proceed faster. This is the basis of enzyme catalysis. An enzyme maximizes binding interactions and binding energy through induced fit. Induced fit also promotes enzyme specificity. Enzymes speed up reactions. They increase the reaction rate. A reaction rate is a change in concentration as a function of time. Reaction rates are influenced by substrate concentration, temperature, pH, and ion concentration. Rates are also influenced by the presence of activators and inhibitors. Many drugs act as competitive inhibitors. Some HIV protease inhibitors act in this way. It's important to understand biological catalysts because they're central to all metabolic reactions. We would not be alive without them.